Good afternoon. I am Prem Paul, and I have the pleasure and honor to be Vice Chancellor for Research and Economic Development. I'd like to welcome you and thank you for joining us today for our Fall Nebraska Lecture, the second of the 2012 Chancellor's Distinguished Lecture Series, especially considering the beautiful fall day. Just delighted to see you here. Today's lecture is being web streamed live, so I also want to welcome those who are joining us via the web. For those of you who use social media, the Twitter hashtag for today's lecture is NebLecture. And Twitter for Prem Paul is PremSPaul. The Nebraska Lectures are an interdisciplinary lecture series designed to foster communication among students and faculty in different academic areas, and among citizens of Lincoln and Nebraska. They are sponsored by the UNL Research Council in cooperation with the Office of the Chancellor and the Office of Research and Economic Development. Research Council is composed of faculty from across many disciplines at UNL. The council solicits nominations for Nebraska lecturers from our faculty on the basis of major recent accomplishments and the lecturer's ability to explain their work to a multidisciplinary audience. Selection as a Nebraska lecturer represents the highest recognition the council can bestow upon an individual faculty member. Well, Research Council also has money. Just in case you're interested, they support several competitive seed grants and programs designed to encourage and enhance research, creative, and scholarly activities for all UNL faculty. So just a little bit about the format today. Following our lecture, Dr. Ken Bloom, my esteemed colleague right here, he's chair of the Research Council and professor of physics and astronomy. If you heard about the Higgs boson, he's part of that team, major discovery. He'll moderate a question and answer session. After that discussion, we'll move to a reception in the Heritage Room just across the hall. I'm also pleased to introduce our sign language interpreter for today's lecture, Thomas Bayer. Thank you very much for your assistance. So let's show our appreciation to Thomas. Usually, Chancellor Perlman attends the lecture and also introduces the speaker, but a scheduling conflict precludes his attendance today. He sends his regrets that he cannot be here. Truth be known, I wanted so badly to introduce our lecturer today that we arranged for Chancellor to go out of town. So it is really indeed, Steve, my distinct pleasure to introduce you. Stephen Benziger is the Nebraska Wheat Growers Presidential Chair and Professor of Agronomy and Horticulture. Dr. Benziger is the first UNL faculty member to hold this endowed chair, which is provided through a partnership between the university and Bayer Crop Science. Since joining UNL's faculty in 1986 as a small grain breeder and geneticist in Nebraska's renowned wheat breeding program, Dr. Benziger has worked to help wheat growers improve their crops and feed the world. His research focuses on improving the agronomic performance and winter hardiness of winter wheat, barley, and triticale, and on developing new breeding methods. He has co-released 42 cultivars and 36 germplasm lines or populations, including 6 new wheat cultivars in the past 5 years. Thanks to the work of Benziger and his colleagues, both past and present, 62% of Nebraska's wheat acres are planted to improved cultivars developed at UNL. His teaching and service activities emphasize graduate education and outreach in plant breeding and genetics. Steve is recognized internationally for his work. He is one of only 2 Americans serving on the boards of trustees of the prestigious International Rice Research Institute. He also has served on numerous scientific boards and panels, including current terms on the scientific advisory boards for CIMMYT's White Yield Initiative and bread wheat. Steve has earned numerous professional honors from both the scientific and agricultural organizations. He's a fellow of the American Association for the Advancement of Science, the Crop Science Society of America, and the American Society of Agronomy. He also is a Nebraska Hall of Agricultural Achievement honoree, Nebraska Agribusiness Club Public Service Award winner, and a recipient of numerous awards from his research and contributions to agriculture. In 2011, he received the Outstanding Research and Creativity Activity Award, or ORCA, the university's top award for research. He chairs the Wheat Genomics Subcommittee for the National Association of Wheat Growers. He also is an honorary professor of China's Ningxia Academy of Agriculture and Forestry Sciences, and the author of more than 200 publications. Steve holds a bachelor's degree from Harvard University and master's and doctoral degrees from Purdue University in plant breeding and genetics. Before joining UNL, he worked on wheat and barley germplasm enhancement for the USDA ARS and on wheat plant growth regulators and biotechnology for Monsanto. Today, he will address the daunting yet exhilarating challenge of feeding the world's growing population. He will explore the possibilities for expanding food production and the role of science in solving these problems. He has titled his lecture, "The Joy of Applied Science While Feeding the World." So please join me in welcoming Dr. Steve Benziger.
First of all, it's a great honor to be asked to give this lecture, and I want to thank Prem for that very kind introduction and the Research Council for the very kind introduction. You've already heard the title of my talk, so the next thing we need to do is to make sure we're in the right room. So I always like to start out with this talk. No man or woman qualifies as a statesman who is entirely ignorant of the problems of wheat. And I think that may have some relevance to our election, but I'm not entirely sure at this point, okay? But it does highlight the importance of crops. And so the topics that I want to mention today will be who we are, the history as it relates to feeding the world, the current challenge, and then I want to pivot to what we do at the University of Nebraska, and then what the future will look like. So those are the topics we want to discuss. Now, the small grains team is large. The university has made a very, very large investment for which, first of all, I am one member, and that I want to acknowledge all of our coworkers, and I'm sure I've missed a few. I've already seen one that I've left off by mistake. We have a large group of geneticists that are with us or with our colleagues in the USDA ARS. Our plant pathology team that works on the diseases of small grains, variety testing, transformation, entomology working on the insects that attack small grains, cropping systems, everything that we breed has to fit into a cropping system. Of course, wheat is predominantly used to make bread or noodles, so therefore we have to have end-use quality. We do a tremendous amount of work with biometry and statistics, which we'll show you how that works towards the end of the talk. We also work with modeling and physiology. As Prem mentioned, we do breed triticale, we do breed barley. All of this, at least my program, is based upon extremely excellent technical support. We do high-throughput phenotyping, which matches our high-throughput genotyping. That's done with CIMMYT, one of the groups at the university. Our funding sources are the state of Nebraska, the USDA through federally competitive grants, as well as through ARS collaborative research. The Nebraska Wheat Board has been one of our longest-term funders, funded me every year that I've been here. We have research and development fees, endowments, and then many of our students are actually supported by their own governments, which is a huge contribution to the program. And what that says is that really I have drunk from wells I did not dig. I have been warmed by fires I did not build. And so while I get to give lectures like this quite often, it's only because that I work with a highly successful team of researchers. And so what you must understand is that agriculture at its core is team research, and failure cannot be an option. Now, the history of feeding the world, basically, for most of recorded history, poverty and food insecurity, that means the inability to feed yourself, were accepted. And even today, probably large segments of the population accepts it still. There are two reasons for that. One was many people believe that it's actually technically impossible to feed everyone. That's totally untrue. We now produce 17% more calories than we did in 1970, even though our population has increased 70% since then. And the second one, which is a little more difficult to understand, is that many people believe that not everyone is worthy of being fed. So everyone living in food security is basically a new idea. Probably came during the Age of Enlightenment, which means it's 300 years old. And so if you look at the 10,000 years of human history, only in the last 300 years have we thought that we could and maybe should feed everyone. Now, to give you a couple of examples of how feeding the world has been looked at in the past, I want to draw your attention to the Irish Potato Famine, 1854 to 1851, 170 years ago. And the person, Charles Edward Trevelyan, I think his name deserves to be remembered, was the head of the Irish Relief. So he would be the head of England's Irish Relief to help make sure Ireland made it through the famine. And his belief was that God had sent the calamity to teach the Irish a lesson, and it must not be too much mitigated. It was a direct stroke of an all-wise and all-merciful providence. That was the traditional or the conventional wisdom during the Irish Potato Famine. Ireland had 8 million people at the time, 1 million died of starvation, and 1 million emigrated. So 1/4 of Ireland in the span of 6 years left the country, either through death or emigration. And that's the person who ran the relief efforts. That was his philosophy. Now, Chesterton, I think, summed it up very well where he said, it was a tragic necessity that the Irish should remember it. This relates to the potato famine. But it was far more tragic that the English forgot it. For he who has forgotten his sin is repeating it incessantly forever. And when you look at people trying to feed the world, I wonder in many times if we are incessantly repeating the sin of thinking it can't be done forever. Now, let's modernize this. Let's bring this up to date. 1968, I was 17 years old. There was a New York Times bestseller. It was written by a National Academy of Science member. And Paul Ehrlich wrote, "The battle to feed all humanity is over. In the 1970s, the world will undergo famines. Hundreds of millions of people are going to starve to death in spite of any crash programs embarked upon now." Okay? That's what he wrote. Now, for your sake, what that meant was if you look at hundreds of millions of people, population was a little bit right around 3 billion. So every 100 million would be 3% of the world's population. When he said hundreds of millions, you're looking at 5 to 10% probably of the world he expected to die of starvation, despite anything we could do at the time. Okay? That was his belief. So what did the conventional wisdom think you should do? Well, they basically thought we should have the lifeboat response. Now, the lifeboat response is that you can't let everybody in the lifeboat, because if you do, then the lifeboat sinks and all perish. So what you do is you get some people in the lifeboat and you leave the rest to their own devices. And so with starvation looming, the only thing left to do was to choose who would be in the lifeboat and who would not be. And it was expected at the time that whole countries would have massive, unavoidable, and because it's unavoidable and you can't do anything, totally acceptable losses of human life. That was 1968. That's 45 years ago, roughly. Okay? Now, I don't want to leave on that pessimistic note because I'm not a pessimist. No one who ever can be a breeder can be a pessimist, okay? With monotonous regularity, apparently competent men have laid down the law about what's technically possible or impossible and have been proved utterly wrong, sometimes while the ink was scarcely dry from their pens. On careful analysis, it appears that these debacles fall into two classes, which I call failure of nerve and failures of imagination. And I think that's very important because while Paul Ehrlich was writing his book, there were two men making a difference in the world we live in. And they are the foundation, with their teams, of the Green Revolution. The Green Revolution in cereals was based upon semi-dwarf cultivars in wheat and rice. The three grains that feed the world are wheat, rice, and corn. Two of the great crops, wheat and rice, were developed with semi-dwarfs. They were given fertilizer and they were given irrigation, and the yields in the poorest parts of the world doubled and trebled almost overnight. So those famines were completely avoided. Okay? Now, Norm Borlaug won the Nobel Peace Prize. By the way, there is no Nobel Prize for agriculture. Norm wanted to change that. He couldn't get it done. So he developed instead the World Food Prize. Norm worked in wheat. Henry Beecher, who was an alumnus of this university, grew up in Wahoo, worked in rice. He was the breeder that released IR8, the great semi-dwarf rice. So he won the World Food Prize, which is what Norm then developed to be something on par with the Nobel laureates. Okay? Now, Norm predicted that those increases would buy us time. He said probably 25 to 30 years, and then if we didn't work with population control, we'd be right back where we started. He was off. Lasted about 40 years. And now we're pretty close to the tipping point again where you have to look at is population and what we can produce in balance. Okay? Now, so the question is, do we need a second green revolution? Now, hunger is the world's greatest solvable problem, and that comes from the World Food Program. And the reason why that's important is for those of you that are interested in this topic, in 2 weeks there's going to be a Herman lecture by a World Food Prize winner who I believe ran the UN food program specifically that works at hunger alleviation. So that's 2 weeks beyond this campus. But food security is based on three things. One is food availability. Sufficient quantities of food available on a consistent basis. That's what agricultural scientists work with. We work on making sure that there is food available. Okay. Second one is food access, having sufficient resources to obtain appropriate foods for a nutritious diet. The third is food use, which is appropriate use based on the knowledge of basic nutrition and care, as well as adequate water and sanitation. So to be food secure, you must have availability, you must have access, and you must know how to use those. Now, what's the current challenge? Well, right now, it's obviously increasing population. We're at 7 billion people. We're going to go to 9 billion people, probably they expect around 2043. And if the trends are right, but it's a long way to predict out, 10 billion by 2083. That's the first concern. The second concern, and this is a good one to be concerned about, is increasing wealth. The 9 billion people in 2043, if their incomes rise, will not eat like 9 billion people today. They're going to eat like 12 billion people today eat. So even if you cap population, the demand on the food system would increase because we're going to eat better and we're going to eat more. Okay? 2010, last figures we have, roughly 925 million people were hungry. Of that, 195 million children were physically stunted due to malnutrition. So they were not growing at the levels that they should. Okay? 129 million are underweight, and 26 million are wasting. They predict that of the 10 million children that die every year, roughly half are due to malnutrition as a related cause. We also have the bio-based economy, and we also have climate change. So that's what we're looking at in the future. Now, in the next 50 years, The rates of gain or the increase in agricultural production that are needed to meet the expected demand are unprecedented. Nowhere in our history have we had that level of increase. And that's even when we had a much smaller level. So 10% improvement on a 1 metric ton per hectare crop is 100 kilograms. 10% on a 3 metric ton per hectare crop is 300 kilograms. So the percentages work against you when you have to have higher percentages and you're at a higher base level, okay? Agriculture will have to play a much greater role in the future than it is now, but that's not dissimilar from what it was in the not too distant past. And one point I think you should understand is that no one in recent history has starved for the lack of food. They've starved for the lack of access. And so as an agricultural scientist, one of the realizations that I've come to is that I can provide the wherewithal technically to feed the world, but if the human heart doesn't change, there will still be starvation. And so what my goal is to provide the technical capabilities to feed the world and hope that the human conscience and heart will decide that that's a worthy goal. Now, what does the program do? In 2010, the Nebraska wheat crop was a little under 4 billion pounds. Its value was about $500 million, okay? Now, as a breeder, I can't take credit for that because a lot of that's production, agriculture, and very highly skilled areas. But the increase due to genetics from 1966 to 2010 was 25%, which means that of that $500 million, roughly $132 million was due to breeding and genetics. Now, at the time, there was 66% market share. Today, it's 62% market share. So if you took 66% of that $132 million, you come up with roughly $87 million of increased revenue or profits at the farm gate for the Nebraska wheat producer. We figured that the university and the USDA, Agricultural Research Service, invests about $4 million a year in our program. So the question I would ask, if you could invest $4 million and get a return of $87 million, would you be happy? If you put your retirement funds in, you put $1 in, you get 20 bucks back, would you be happy? You can be happy. It's okay. It's okay. Now, I like people making money. I like our farmers being more competitive. I like them being more productive. I like them competing anywhere in the world, okay? But the real point on this is that's not what got me into agriculture, okay? If you want to know why I got into agriculture, it's this middle column. You take that same 4 billion pounds of wheat, it feeds about a little over 20 million Americans their annual consumption of wheat. Again, a breeder can't take credit for all of that, but 25% of that's due to genetics, which is a little over 5 million people get all the wheat they eat from the Nebraska crop based on genetics. Again, we can't take credit for all of that because only 66% of the crop is from our varieties, but about 3.3 million people get fed on the basis of what the University of Nebraska and the USDA invests in wheat. Now, for your sake, that's only in Nebraska. Our varieties are grown in Kansas, they're grown in Colorado, they're grown in Wyoming, they're grown in South Dakota, they're grown in North Dakota, and they're grown in Minnesota. So the actual value is much higher than the $87 million. The actual number of people is much higher, but because this is Nebraska and we work for Nebraska, those are the figures we've given you. Now, for those of you that are familiar with this phrase, give us this day our daily bread, I want to come back to the importance of agriculture. Few scientists think of agriculture as the chief or model science. Many indeed do not consider it a science at all. Yet it was the first science that makes all human life possible. And it may well be that before the century is over, the success or failure of science as a whole will be judged by the success or failure of agriculture. It was written in 1974. That's when the Green Revolution was just beginning to take off, and they weren't sure if we would make it to the 21st century. We made it because of the success of agriculture. How we will do at the end of the 21st century remains to be seen. That has yet to be written. A good friend of mine is Jan Leach. She's a plant pathologist, and she was flying home one day, and she sat next to a doctor from the World Health Organization. And he went on about all the great things that the medical field was doing. They were curing malaria with their mosquito nets. They were vaccinating people against polio and smallpox. They were giving nutrition supplements. They were doing all kinds of wonderful things. Hundreds of millions of people were better off because of that. Then he looked over to this young woman and said, "And so what do you do?" And she said, "Well, I'm an agricultural scientist, and my job is to feed all those people whose lives you save." And what she didn't say is we also feed all the lives they don't save. Okay? Now, let's talk about what we do. Okay, so we've laid out the picture, we've laid out the challenges. Now let's talk about what breeding does. There are three things that every plant breeder works with. The first one is to introduce variation. That takes 1 to 2 years. Then we allow all that variation to segregate out. Okay, that's to separate itself. And then we make our selections of what's the best types of plants that we can find. That takes years 3 to year 6. And then after that, we evaluate the best selections to make sure they have merit so we can release them to growers and they can be more profitable, more productive, and feed the world better than what they could do before. That's year 7 to year 12. So 12 years from start to finish. And the reason why I bring that up is I'm 61 years old. I make 1,000 crosses this year. I will not see the varieties from those 1,000 crosses. I doubt I'll be working at 73, okay? So I'm making it for my successor the same way as my predecessor made it for me. That's the continuity that leads in agricultural research. Now, if you look at introducing variation, the way we normally do it is through a cross, okay? And what you see, that's us pulling out the anthers. That's the male parts. The plants have both male and female parts. We leave the female parts. I don't know if you can see these feathery things. That's the stigma. That's the female part. We will hand pollinate that, and from that we'll get a seed. The second stage is the segregation and selection. Every Nebraska variety has to have 4 characteristics. The first one is it has to survive the winter. It's perfectly acceptable if it dies as a plot in my field. It's totally unacceptable if it dies in a farmer's field. It means we haven't done our job. For our pathologist friends, it must have resistance to stem rust. It's the black rust. It's the biblical plague of Egypt, one of the 7 plagues of Moses. Okay? It has to stand in the field, have good yield, have good test weight, be harvested, fit the cropping rotation. And lastly, it must make a good loaf of bread. This is an indication of bread making quality on a micro test. We do a lot of things at Lincoln and Meade. They're close. We have a lot of small plots. We use small plot equipment. This is the mixograph. What I like about this is it takes a little more than an ounce of seed to make enough flour to determine whether or not it can be advanced and may have potential for making a loaf of bread. Ideally, a mill would want this kind of curd from the mixograph, which comes from here. We generally try to breed for this, the reason being that if you have heat or if you have drought, do you ever have that in Nebraska? Ever? What happens is this curve tends to become more like this. So we always give the mills slightly better than what they want, so in the bad years, they get exactly what they want, rather than giving them exactly what they want in the bad years, giving them something that's unacceptable. So we always build in a little margin of environmental error. Now the last phase is the evaluation and release. These are our testing sites. And first of all, for those who are unfamiliar with Nebraska, from Omaha to Scottsbluff, there's as much climatic variation as there is from Omaha to Washington, D.C. Okay? So everything we live with is based on environmental variation. If you look at this, in the eastern corner, where about 1/3 of our wheat is grown, this has roughly 750 millimeters, 30 inches of rainfall, 300 meters above sea level, roughly 900 feet above sea level. This is where the best corn and soybean ground is under rain-fed conditions. You go to the central, this becomes wheat, and you follow it with corn if there's moisture, or you follow it with sorghum if there's not enough moisture. This has probably 500 millimeters or 20 inches of rainfall. And then there's a third of our wheat in this area. There's a third of our wheat in the west. This, you're looking at 250 millimeters of rainfall, probably close to 1,300 meters or 4,000 feet of elevation. Okay? Now, if you look at our testing sites, Lincoln, Mead is our winter hardiness testing site. If something survives at Mead, it will survive anywhere in Nebraska. Very critical site. Clay Center. We have McCook, which is a farmer's field, North Platte, which is a university farm. These three are all university farms, as is the Haskell Lab. Sydney is a university farm, it's in the west, and Alliance is on a farmer's field. If you look at it, we have basically the winter killing site, and we have 2 sites in eastern Nebraska, 2 sites in southwestern Nebraska, 2 sites in western Nebraska. We do that because we lose sites to hail, we lose sites to heat, we lose sites to drought, and our goal is to never lose both sites in one of the 3 main ecozones that we have to produce wheat in. So that's why we do that. Now, the other thing that's very important, which makes our job very nice, is if you have a wheat that does well at Lincoln, does well at North Platte, and does well at Alliance, you'll own the northern Great Plains. Why? Lincoln is very similar to eastern Kansas. North Platte is very similar to eastern Colorado, western Kansas. Alliance is very similar to Wyoming, South Dakota, North Dakota, and Montana. So if a line does well at those 3 sites, it'll do everywhere from Nebraska north. It's an excellent ability to select. Our breeding program, we go to much larger plots. Our combines are all computerized now. We do much larger milling. That's an experimental mill. This is what a commercial flour looks like making a loaf of bread. This is an experimental line that will never see the light of day. Basically, that's better for bricks than for bread. And this is an experimental line that has very, very good seed quality. And this is in our baking laboratory. Now, so I've shown you what we've done in the past. But the thing you have to understand is that a good past is positively dangerous if it makes us content with the present and unprepared for the future. And so the point I want to make here is twofold. Number one, how do you grow profitable wheat? It helps if you got an oil field. Okay? That helps. But the second thing is, even though we have been successful in the past, doesn't mean you're going to be successful in the future. So now we're going to pivot a little bit and talk about where we are in the future. This is a great quote by Louis Pasteur, the microbiologist. No, 1,000 times no, there does not exist a category of science to which one can give the name applied science. There are science and the applications of science bound together as the fruit to the tree which bears it. So the title I gave you is a little bit of a trick, because most people forget that science can have an impact and can make a difference. I wanted to highlight that. But at its core, plant breeding is a science, and it has the same curiosity-driven types of things as you'd find in any other science. And so the theme for any plant breeder is simply this: we shall not cease from exploration, and the end of all our exploring will be to arrive where we started and know the place for the first time. Yes, we can be successful doing things the way we have, but we're not going to be as successful as we could be if we can understand what we do better. So that's our future, is better understanding and laying a better foundation for what we want to have in the future. So let's go back and look at the 3 phases of plant breeding and how they're changing. When we introduce variation, transgenes. Now, I don't call them genetically modified organisms. That term infuriates me. Everything I breed is genetically modified. So when you call it genetically modified organisms, we've lived with those for 10,000 years. Transgenic organisms are relatively recent, and that's the term we're going to use. We're going to use wide hybrids so we can mine genes from wild relatives we didn't have access to in the past, and we're going to use mutations. Transgenic crops, to me, is the first of 2 frontiers left for wheat, the second one being hybrids. We won't spend much time on that, but this one is very important. And I give you an example, which to me is, I just think, very clever. North American maize is attacked by the western corn rootworm. Now, the western corn rootworm has a biological control agent called a nematode. The nematode will attack and basically kill the western corn rootworm. Ancient maize had a signal, which was a sesquiterpene olefin. So when the corn rootworm fed on the roots, the maize would release this signal. The signal would then say, I'm being fed on, there's food here. The nematode would be attracted to the signal and eat the corn rootworm. Okay? Unfortunately, as we bred modern maize, that signal got lost. Okay? So one of the things that happened is, how do you put the signal back in? Because the biological control still works very, very well. Well, you can do it two ways. You can cross back to the ancient maize. You can do that. Problem with that is you have a lot of genes you no longer want because you got rid of them as you had this great history of breeding. Or two, you can transform the maize with an enzyme that'll make that signal and put it under a promoter that will be turned on when you need to have it. So in this case, the research team found that oregano. Now, anybody familiar with oregano? Spice that's in almost every kitchen. You consume it, right? No problem. They found that it had the enzyme that made the sesquiterpene olefin. They regulated it or put it under a promoter so that when the corn rootworm fed on the roots, the signal was released. The nematode said, buffet, come on down, ate the corn rootworms, right? What was the end result? 60% of the rootworms that in the uncontrolled, which would be the non-transformed plants that adults would have left, they're the beetles, okay, the rootworm is the larva, were killed. So you could reduce the effect of the insect feeding by 60%. Not bad for taking a gene out of oregano and putting it into maize. Now the reason why I bring that up is you're going to see the golden rice will probably be released in 2013, 2014. Nobody thinks we can cross oregano with maize. Nobody thinks you can cross daffodils with rice, but you can make huge differences due to this type of technology. Variation through mutations. The little yellow is a half a chromosome arm that's probably grown on hundreds of millions of acres of wheat. It comes from rye, gives you disease resistance and drought and heat stress tolerance. Okay? The other field, almost all wheat is susceptible to one of the herbicides that's commonly used in other crops. With a mutation, you can make it resistant. That's normal wheat. That's the wheat with mutation. That's grown on hundreds of thousands of acres of wheat in the Great Plains. Simple mutation. We do a lot of molecular markers. The first use we use is to understand how to select parents. Where do we get this genetic diversity from? In 1870, the wheats that were grown in Nebraska were spring wheats. It was only when the Mennonites, which had fled Germany because they were going to be forced into the army, had moved to Russia because the Tsar said, you could not be in the army, we just want good farmers. He died, a new Tsar came, said, we want you in the armies. They left Russia to come to Kansas. They brought with them their wheats, and the wheat they brought was Turkey Red. Now, Turkey Red wheat was what they call a steppe wheat, which meant that it could take heat, it could take drought, and it could take fierce winters. Sound familiar? Okay. The wheats that were grown in Nebraska prior to that time were English wheats. They didn't have hot summers, they didn't have cold winters, and they didn't have very much drought or heat. Okay? And so that's what was grown in Nebraska. Now, what we then said is this is interesting. 100 years of breeding in Nebraska, 100 years of breeding in Turkey. Geez, I wonder if we could go back to Turkey and get some genes that maybe we missed. So what we did is we looked at all the wheats from Turkey, historical to modern. They're the ones in black. We looked at our Nebraska wheats. They're the ones in blue. Okay? And we decided to see how they related using molecular markers to see if we could go back and find new sources. By the way, this is a monument in Newton, Kansas to represent the Mennonite farmers who brought in the Turkey Red wheat. What we found is here's Turkey. This is the first great wheat released by the University of Nebraska in 1933, Cheyenne, highly related to Turkey. But interestingly, one of our great wheats released in 1984, Siouxland, is very similar to the original Turkey. And you can see this cluster, this is a cluster diagram, where you have the Turkish wheats in black, very similarly related to the blue wheats, which are ours. Right now, the most popular wheat in Nebraska is this one, which is Overland. It belongs in this cluster, which has a series of very, very good Nebraska wheats and a few Turkish wheats. So now we know where to go back to get our germplasm to improve our own wheats. Now, the other thing I should mention to you about Turkey wheat is I mentioned the Green Revolution. It was based on a parent from Japan, Norin 10, and it was based on a parent from Washington State, Brever. Norin 10 from Japan was a cross between Fultz, that's an American wheat, Daruma, which is the source of the semi-dwarfing wheats, and Turkey Red. I'm always amazed at the germplasm exchange that must have occurred in the 1930s and 40s, and how the bloodlines that led to Turkey Red have led to the global Green Revolution. What's happening in our second phase, segregation and selection? Much better assays, much better genomics and genetics. High throughput genotyping is making a huge change. And of course, double haploidy. Just looking at field plot technique, one of the things that's really revolutionizing and making our life much easier is global positioning satellites that are controlling our tractors and our drills. So that now you will plant perfectly straight rows and you can trip your drill basically by where the satellite tells you to trip it. First year we tried that, you can see how nicely our plots lined up. We're even better now, we've taken out even that little bit of stagger. And it means that we go into a field, we set up our antenna, we plant 400 to 500 plots an hour, and we do that without ever having to crosshatch and without ever having to have highly skilled technologists watching that line to make sure they drop at the exact same spot, both going up and coming back. It's a huge advantage, and it makes our yield trials much more precise. Most of the early generations is done with visual selection. The human eye is good. The human brain is a magnificent computer, but there's many spectral images that we can't see. So we're now going through very high throughput phenotyping using Canopy spectral reflectance. In this case, we have sensors that are over the plots. The data is transported to a backpack. That backpack data is transferred to a computer, which can be uploaded to a satellite to our servers. 53 million data points from one experiment with 1,300 plots, looking at nitrogen use efficiency and looking at wavelengths that we can't even see, and hoping that we can get signals to help us understand what's going on. Now, that's using ground-based things. You can also use blimps to carry those sensors. This is the International Maize and Wheat Center, the global lead for wheat and maize breeding. They use blimps. They run it over their plots, and they can get hundreds of plots in every image. Really nice, okay? The problem with it is that they're not stable in the wind, which puts us at a disadvantage because we often have wind. But there are some really cool alternative uses for blimps. Shameless promotion of your own seminar, okay? Highlighting the distinguished leadership of the university, you know? Don't you think they need a blimp? I mean, really, when it comes right down to it. Also with CIMMYT, we're now using aerial flyovers, okay? And the study that you saw that student in is here and here. And what's interesting is with an aerial flyover, it's less time to record all the data from these plots than it takes to fly from Lincoln to Meade, where the experiment is. That's high throughput phenotyping. And the other part is that you're doing it at the same time, so you don't have a cloud fly over and suddenly change all your data, okay? What's also interesting is my main breeding nursery is in this field. Now it extends beyond here, but you can see I can do this 1,300 plots and I can do the 5,000 plots there in almost no time at all. Again, the bioinformatics on that are going to be scary, but it's a great way of really getting data that we never had access to. Not everything has to be done from the air. This is a kernel sorter. Anything that's done with near-infrared reflectance, you can separate red kernels from white kernels. That's the way markets are separated in the world. You can separate diseased kernels from healthy kernels. You can separate high protein from low protein kernel. Anything you can do with the NAR, in theory, you can do with these kinds of equipment. This is a prototype. The one we have in our laboratory can literally do hundreds of thousands of kernels a day. Doesn't do all the things this one can do, but it can do what I need. Of course, molecular markers are extremely important. And the key part on this is we didn't understand what yield was. We knew how to measure it, but we didn't understand the genetics of yield. And what this shows is that there are major genes involved in yield. This is a chromosome. These are the markers right here that happen to bracket a major gene. Now in blue, it's the average effect. Each one of these is a different environment. In 3 environments, statistically significant effects on yield, 15% yield enhancement. That's significant. This one, not statistically significant, but clearly the gene, and in 2 environments, no effect at all. So we now begin to understand how yield works over multiple environments. Again, Nebraska has more environmental variation than just about any other state, but we also can understand how yield is built. What's interesting is if you take the lines that have the allele for higher yield, which are in the purple squares, and you compare it to the lines that have the lower yielding allele, which is in the blue diamonds, you can see that and these happen to be low yielding environments, these happen to be high yielding environments. You can see in the high yielding environments you get a very distinct advantage, and in the low yielding environments you don't get an advantage, but you don't get hurt either. So it means there's no downside whatsoever of using this allele. The worst it can do is be no help. The best it can do is a 15% yield advantage. So that becomes a huge capability for trying to improve crops. Now, what do single genes do? As Prem mentioned, among other things, I'm on the board of trustees for the International Rice Research Institute. I'm out in Orissa, India, one of the poorest parts of India. I'm looking at these trees and I see, dang, they're the weirdest looking trees I've ever seen because there's all these roots hanging down from the trees. That's 8 feet above the ground. They're not roots. What that is in the monsoons there was a flood. And the flood was 8 feet deep, and it took all the litter from the fields, all the reeds, all the grasses, all the branches, all the twigs, and it hung them up in the trees, and they just haven't degraded and fallen down. So that's the high water mark of the flood. Now, this is an area that grows a lot of rice. Rice is grown in water. Everybody knows that. But what you don't probably know is that rice can drown. If you cover the rice plant completely to the top, it will drown. So that's one of the major problems when you have monsoons is you can drown your rice. But they found a single gene called sub1, which is submergence tolerance, and put it into a major variety swarma. And so this field is one that had the submergence tolerance. It survived that flood, can allow rice to be underwater for 7 to 14 days. This field, they didn't have the submergence tolerance in. All the rice died. Okay? Now, it shows you what a single gene can do. Now, I also want you not to think that this farmer was foolish. There's just not enough seed of the sub-1 varieties yet. They're increasing the seed, and when there is enough seed of that, everybody will grow it, but there just wasn't enough. Okay? This is me with my new best friends. Frankly, I love growers. They're the same worldwide. They all have the same concerns, and they all have the same interests. Again, we talked a little bit about using markers to find single genes. We're looking now to use multiple high-throughput genotyping to understand yield much better. This is a plot showing how things cluster together. It's done by one of my students. 280 lines are the gateway to all the varieties we release. Using, in this case, about 2,000 markers, we were able to cluster lines into one group, a third group, or second group, and a third group, or 1, 2, and 3, okay? That's very important because when we made our selections, we were worried, are we eliminating, due to the selection year, some of the variation we may want to keep? And it ended up that these green dots, which were the ones we selected, represented all 3 of our clusters. So even though we're selecting, which means you're narrowing your diversity, we were keeping the main set of diversity the whole time so we could understand our diversity better than we ever could before. What's the last thing? Looking at the evaluation and release. And I guess what I should say is much better work on cropping systems, much better work with biometry, much better work with modeling. This is what drought looks like. And I want you to think about how do you put a yield trial into a field like this, where it doesn't matter what you planted here, it wasn't going to emerge, or here where only half of it emerged. So how do you take care of that kind of field variation? Same thing happens with the winter. Anything that has snow on top of it will survive the winter better than anything that doesn't, because snow works as an insulator, okay? Anything that has snow on top of it will have a better start in the spring, because when that snow melts, there's more water on that plot. So how do you go into here and try to compare this plot that's heavily covered with snow with this plot that has just a little bit of it? You use very sophisticated statistics. I don't know if we have any of our statisticians with us here, but that's a huge part of modern biometry. Now, there's three things that every breeder really enjoys, okay? The three joys. The first is when you make the cross, because that's when all the creativity goes in and you think about, if I can get this trait from this line and these traits from that line, I can come up with something that's really good, okay? The second is when you're out in the field and you're looking at your selections. And when you see a line and you know it's going to be a variety, you will know where you were in the field, you'll know what time of day it was, you will know whether it was cloudy or windy, you'll remember it. And the last is when that variety is grown on millions of acres, and you remember when you held all the seed of it in the palm of your hand. That's what makes a breeder excited, okay? And my message to you is never forget what you hold in the palm of your hand. If you can imagine it, you can grasp it. That's what breeding's all about. So there's an old African proverb, which is, if you want to go fast, go alone. If you want to go far, go together. And I say that because I want you to remember that what I've presented to you is part of a team research. We want to go far, and we do it because we go together. There are 9 billion people to feed, and we all need to work together. With that, I want to thank you for your attendance, but I also want to thank the University of Nebraska for allowing me and my colleagues to do the work we love and to do the kinds of things that make us excited. Thank you very much.
Okay, thank you, Steve, for that very nice description of how research and research done at Nebraska specifically can really have an impact on the world. I'm Ken Bloom. I'm the chair of the University Research Council, and I'll be moderating our discussion this afternoon. Let's not forget, we do have people watching over the web, so if you have a question, we'd ask you to please come to the microphone so that people on the web can hear us also. So we'll open the floor for questions.
My question's regarding the submergent rice.
Mm-hmm.
Could you elaborate process how this was created, and also is it applicable in Southeast Asia as well as South Asia?
Okay. The question, and I hope everybody heard it, was about sub-1. How it was found, and it's like many traits that are found in agriculture. It existed in rice, okay? There were rices that were known to be more dry, or more flood tolerant than others, but nobody understood the process. They didn't know if it was a single gene or if it was like 100 genes, which would be almost impossible to breed. And so what they did is they made crosses. They put molecular markers on it, which are like signposts on the DNA, and they found at one address there was something that made a difference in submergence tolerance. So they were able to say it's not a neighborhood, it's not a city, it's basically a single house in that neighborhood that controlled this emergence tolerance, okay? So that's how they found it. They used it through basically molecular mapping and looking at segregating populations. Okay. Now, the question then that you asked was, is this trait useful in South Asia or Southeast Asia as well as South Asia? And the answer is it appears to work everywhere. Everywhere that rice is flooded and the rice is totally submerged, this trait will work. Okay? And in fact, all of the early development work was not done in India. It was done in the Philippines, where Erie's headquarters are. So that's where they did the initial studies. So they found the gene, probably, as I recall, in South Asia rices, but did the validation and the testing in Southeast Asia, and now it can be used globally. You bet.
And it's okay if you want to form a queue, then we'll get people through.
Sticking with the submergence tolerance rice, you mentioned that one farmer with the flooded field was not being foolish. There just wasn't enough seed. When there was enough seed, everyone would plant it. As a plant pathologist, that is a terrifying statement. So I'm assuming there are efforts underway to ensure that that trait gets into multiple different cultivars and to ensure that that we're not encouraging widespread monoculture to so we don't have an entire continent wiped out by a single disease that really likes that line.
Yeah, Heather, that's a great question, and I appreciate your plant pathology orientation on that. And the answer is the sub-1 gene is being back-crossed into numerous rice cultivars, okay? And specifically to avoid the concept of genetic vulnerability where one disease could take out everything. The gene itself appears to have no relationship to any known disease, so the gene itself will not be a problem. But fortunately, as with rice, there are many adapted cultivars, and so to have it actually work properly, you have to back-cross it into every one of the adapted cultivars, and that would lead to the genetic diversity that you in fact need to avoid the genetic vulnerability of massive epidemics and catastrophes.
Thank you very much for a fantastic lecture, but also for all the great work you've done. My question is really related to that, is that you mentioned that you and your colleagues takes a team approach. And would you mind addressing where are we preparing sufficient pipeline with the type of skills that are needed so we are able to address issues into the next century?
Okay. There's really two ways of answering that question, Prem, and thank you for the question. The first one would be, are we preparing our students to be the type of breeders that the next generation would be? And I think the answer to that is clearly yes. And I'm very proud to be part of the University of Nebraska because we have invested heavily in agriculture. And the type of equipment we're using here, which we consider state-of-the-art, is routine in industry, okay? So what we're seeing is that 75 to 80% of our graduates work in industry, and the question is, are we training them for the jobs they'll have in the future? And the answer is yes, because we've invested in the type of technology so they will not be unfamiliar when they go work for a Pioneer, a Syngenta, a Bear Crop Science, a Lima-Grain, whatever. So that's the first part. I think we're doing very well on educating our students. Now, the second part, which I feel equally very proud of, is that literally that last comment about if you want to go far, go together. And one of the things that I appreciate is that the University of Nebraska has invested in the type of collegial relationships which will allow us, again, to educate our students in a manner that is second to none. So if you think about Innovation Campus, okay, the reason why I'm most excited about that is it's great to bring companies here. That's fine. But I think that public-private partnership is what's going to be the future not only of universities with private industry, but the future of education as we try to make sure that our students can have the jobs academically or private sector that they want to have. So I view the Innovation Campus as a huge step forward. The agreement we have with Bayer Crop Science, I think, is nothing short of phenomenal. It's to me, the dream of a lifetime. I don't know how many of my Bayer friends are listening, and you know, probably I'm supposed to talk about, you know, keep it modest and all that stuff, but I just think it's a dream of a lifetime. I mean, it's one of the most coolest things that I've ever seen happen. And I think, again, it's helping our students, it's helping our program, it's helping industry, and that goodwill and that good-naturedness that we have only leads to better things in the future. So, and I hope, and we're going to have a stakeholders meeting in the end of November, where all of the team can get together and talk about how we want to invest in our future. And I think that's huge. And these types of agreements have helped us have the resources to do that.
So I'll ask a question, which is, well, it's a question I know how to ask because it's closer to my field. I mean, you mentioned, you know, the case of having 53 million data points. I mean, I presume this is relatively new for agricultural research to have such large data sets in that way that you're going to have to process in some way and get something out of. How is the ability to collect data about your varieties changing your field in terms both of the opportunities that it might present, but also the challenges of dealing with this new way of working?
Okay. Well, first of all, I in this audience, I don't want to make it sound like that 53 million data points are new to agriculture. I mean, people like Mike Fromm that are in the audience, and where is, I don't know if Jeff Mauer's in the audience or now, but they generate 53 million data points in their sequencing, you know, probably fairly quickly. Would that be fair, Mike? Yeah, very quickly, okay? So that's not an issue there. The difficulty or the interesting part is that we're trying to tie phenotypic data, which has been, you know, at one time we could phenotype more than we could genotype. Now we can genotype unbelievably. I mean, you know,
DNA sequencing.
DNA sequencing is just extraordinary. I mean, we get 45,000 data points per line on 300 lines, and it costs us, I think, about $7,500, $15,000, something like that. It's an unbelievable amount. So now, those of us that have never had that have got to figure out how to do it. Yeah. Well, we go talk to bioinformaticists, people that Mike and Jeff Mauer have worked with and others, and we do that. But then the question is, how do you tie that to 53 million spectral points to find out which gene fits with which spectral? And so that's an area of bioinformatics which I think is evolving, and we need a lot of help on. And of course, the other thing is that, you know, this is not the kind of thing you do with an Excel spreadsheet, which, you know, my generation grew up on. You know, an Excel spreadsheet. I remember the first, you know, well, heck, I remember the computer I did my PhD on, I think, had 4K memory. I think that's right, you know. And so now we're looking at these things. So clearly, you know, those of us are not educated to do that. We're going to need a lot of help. And then the other thing is, how do you get high quality? The sequencing data is probably the highest quality data you can get. You're out in the field, and I've shown you that field variation. How do you make that data equally high quality? That's what becomes difficult. And that's where our biometricians and bioinformaticists have got to help us.
Other questions? Don't be shy. Okay, well, if not, then thank you again, Stephen.
Thank you.
Steve, as you know, that we generally make a poster of the to advertise the lecture, and we have a framed copy of this poster with a handsome young man in it. So we'd like you to put it in your office, one copy is going to be in our research building. So great. Anytime you want to come and look at that, and you want to look at the young man, you're most welcome to. But thank you very, very much, and keep up the great work.
Thank you, Trevor. Thank you. Thank you.